Specific immunity protects against specific pathogens. It develops over your lifetime. It is the coordinated activity of both T cells and B cells that provides these specific defenses. T cells are responsible for cell-mediated immunity, which means the cells directly contact the invader to destroy it. And B cells provide antibody-mediated immunity, and the antibodies attack the invader and cause usually a clumping type of reaction. So there are two types of specific immunity, active and passive. Active means you produce your own antibodies. Passive means you receive antibodies from another source. And both active and passive can be naturally acquired or artificially acquired. So let's look here at active immunity. Active immunity means you make your own antibodies. You could make them naturally if you contract the disease, or you could make them artificially if you're given a vaccine. With passive immunity, you're receiving those antibodies from another source. You could do this naturally if you were a newborn from your mother's milk, or you could do it artificially with an injection of antibodies that have been taken from someone's plasma. There are certain properties of specific immunity. Number one, specificity. Each T cell or B cell is attracted to one specific antigen. That's it. So each antigen can have its own T cell or B cell that responds to it. It's versatile. There are millions of different lymphocyte populations, and each is sensitive to a different antigen. When activated, the lymphocyte divides, producing more of exactly the same type that responds to that specific antigen. This is called a clone. There is memory. Once the lymphocytes have been activated, they produce cells that will remain in circulation in the body, memory cells, that will respond much faster the second time you're exposed to the disease. And there's tolerance. That means that there is no attack on our own cells. So again, exposure to an antigen activates phagocytes. And what's going to happen is the T cells will start a cell-mediated defense that is directly attacking the invader. And the B cells will initiate antibody-mediated defense that is producing antibodies that will attack the invader. But in both cases, the first step is antigen presentation. So an antigen-presenting cell, APC, is a specialized cell, a macrophage. The macrophage is going to engulf the pathogen and then digest it, but retain a certain antigenic piece, a certain protein. And it's going to attach that protein, the antigen, to another of its own proteins and display it on the plasma membrane. So let's look at a picture of that. So here's the APC, the phagocytic antigen-presenting cell. It engulfs the bacterial cell. Lysosomes fuse to it, digest it. So let's look at one of these little red dots, a particular antigen. Certain proteins produced in the rough ER are going to bind to that protein and exit the cell and attach to the cell surface and present that antigen like a trophy. This antigen piece is being displayed on the membrane. So different things happen with different invaders. If you're invaded by a bacterial cell, first the macrophages are going to attack it, and then there's going to be antigen presentation by that macrophage. A virus, well, viruses are really small and too small to be affected by a macrophage. So the macrophage might have contact with a virus-infected cell and then use antigen presentation. The cells will also release interferons that affect the surrounding cells, preventing them from becoming infected by virus. Memory cells are not important when you're first infected by a pathogen, but they are produced to that specific pathogen, and they will respond much, much faster the second time. So we have cytotoxic T cells and helper T cells. We have memory of both. And then we have memory B cells that will produce antibodies so much faster the second time. Which of the following functions is associated with B lymphocytes? Antibody production. A B cell differentiates into a plasma cell, and the plasma cell is the antibody factory. It just pumps out hundreds and thousands of antibodies. Which of the following is not one of the properties of specific immunity? Innate. Innate means it's inborn, so it's non-specific. There's a little bit too much detail about antibodies. Instead of reading about that detail, we'll look at this antibody here. It's got 2 heavy chains, the purple ones, and 2 light chains. Really, it's a Y-shaped protein. And the Y-shaped protein can bind to the antigen to form an antigen-antibody complex. Sometimes the antigen is too small, so it binds to a carrier molecule, and then the antibody can attach to it. So in this picture here, it's not a very good picture because the antibody really is much smaller than the antigen. So it's maybe more like this, where you get a lot of antibodies binding to one antigen, and it causes this antigen. It makes it unable to infect our cells and may allow it to be engulfed by a macrophage much faster. It may cause clumping of cells. So there's 5 classes of antibodies, and an antibody is called an immunoglobulin. Remember when we were talking about the plasma proteins, we had globulins. Antibodies are globulins. They're immunoglobulins. And we have IgG, IgE, IgD, IgM, and IgA. And here we go. It's just a Y-shaped protein. And IgG, 80% of all antibodies are IgG, and they attack just about anything. Viruses, bacteria, bacterial toxins. IgE is more for allergies and inflammation. It attaches to basophils and mast cells, and mast cells contain histamine. When they release histamine, it kind of speeds up inflammation. IgD, yeah, I don't think you need to to know the different purposes of each one of these. I would say know that IgG is the most common one, and IgA is the one. I don't know if you've ever heard of the grocery store, the IGA, but I used to go to the IGA to get milk. Well, IgA is found in milk, saliva, mucus, tears. So the primary response is the initial response to exposure. It's basically slow. The antigen activates the B cells. The B cells differentiate into plasma cells, and they make antibodies that are specific against that antigen. You can count the antibody titer. It peaks 1 to 2 weeks after exposure and then declines if it's no longer exposed to the antigen. Well, the secondary response is when you're exposed to that disease or antigen a second time. And this response is more extensive, lasts longer, much faster, higher antibody titer more rapidly. And this is because of the memory cells. And memory cells can last for 20 years or more. So let's look at the primary response to IgG, which is the most common one, in IgM. Look at the peak at 2 weeks. Okay. Well, 2 weeks after you've been exposed to a disease, you've gotten sick already. For the most part, usually the incubation period is less than 2 weeks. Although with coronavirus, it's, a few days to 14 days. But that's unusual. Usually it's like 24 hours, 48 hours, So the response is slow, but you don't die. The second time you're exposed to it, look at the peak here of IgM after 1 week. And look how fast IgG is increasing. So that secondary response is much greater and faster. So you do not get the disease. Which of the following functions is associated with B lymphocytes? Antibody production. Which antibody class is secreted first and forms a large structure that can agglutinate red blood cells? IgM. What is the function of a macrophage? Display antigenic fragments to activate T lymphocytes. So let's see what happens. How does the antibody actually kill the invader? So briefly, we'll talk about each one of these ways. So neutralization, bacteria and viruses must bind to the plasma membrane of body cells before they can enter the cells. Antibodies may attach to the binding site on bacteria or toxins, and it neutralizes the binding ability. So if there's no binding sites available, then it can't enter the cell. It can prevent pathogen adhesion. Antibodies are in saliva, mucus, tears, and perspiration, and create a barrier. Makes it difficult for pathogens to attach to our body surface. Activate complement. Well, we're not going to learn much about that, but it's proteins in our blood, and binding to the antigen changes the antibody molecule shape, and this exposed area will bind to complement proteins, activating an entire system called the complement system. Opsonization, coating of antibodies and complement makes surfaces of bacteria less slick, so phagocytes can bind more easily. So basically, opsonization makes it easier to phagocytize the pathogen. Attraction of phagocytes. Neutrophils, eosinophils, and macrophages are attracted to antigens that are coated with antibodies. Stimulate inflammation, which antibodies can stimulate the release of heparin and histamine from basophils and mast cells. Basophil is a type of white blood cell, and we saw mast cells in our connective tissue, and it promotes the inflammatory response. Agglutination, antibodies can bind to the antigenic determinant site on adjacent antigens and create an immune complex. It's just a kind of a clumping reaction called agglutination. Which of the following does not bind to antibodies? The helper T lymphocyte. Which antibody mechanism prevents the binding of viruses and bacterial toxins to body cells? Neutralization.